Good afternoon, and thank you very much indeed for having me. I'm just going to read the poem on this slide because I realise that the text doesn't project very well. The seed. How does it know, this little seed, if it is to grow to a flower or a weed? If it is to be a vine or shoot or grow to a tree with a long, deep root? A seed is so small, where do you suppose it stores up all the things it knows? So just as Clive gave a very kind introduction, I'm here in my capacity as Dr. Alison Mail, consultant in clinical genetics. But just to set expectations, before I tell you what I do, I would like to make it clear what I don't do, what I am not. And the first thing is, I am not, and never have been, an evolutionary biologist. In fact, I didn't even do biology A-level, so I've never studied evolution. So if you were hoping to have a debate about evolution today, I'm afraid you're going to be really disappointed. But if that's something you're interested in, can I recommend a book written by a hero of mine called Francis Collins, who is the leader of the Human Genome Project, a proud scientist and a very, very proud Christian, an American gentleman who's very knowledgeable about lots of things. And so if that's what you're hoping for, not going to give it to you, but you can find it here. I am also not a researcher. I don't have a PhD and never ever had the desire to get one. I work with a lot of researchers. I recruit patients to research projects, but I will never have a syndrome named after me. I do not do research. And I am not a clinical scientist. I myself do not do genetic tests in the lab. I work very closely with the scientists in my team who do, and I talk to them about those results, but I am not clever enough to do the tests myself. What I am is a doctor, a medical doctor, in the medical speciality of clinical genetics. I trained at St. George's Medical School in Tooting, and then I did my postgraduate basic medical training. You'll be pleased to know that basic medical training comes after medical school, after you're a doctor. You do your basic medical training, and I did mine in pediatrics. And then I did a brief job for a few years in something called neuromuscular disorders, so that's disorders of the nerves and muscles. And then I went into my training in clinical genetics at Guy's way back in 1997. And because I had children, I worked part-time, so it took me 7 years to train in genetics. So I actually got my consultant job in Great Ormond Street in 2004, which was 14 years after I qualified as a doctor. So, you know, medical training is quite long. What is clinical genetics? Clinical genetics is a medical specialty that provides a diagnostic service and genetic counselling for individuals or families with or at risk of conditions that may have a genetic basis. So back to our little poem about the tree or the seed. It comes from this book, which was a poetry book that my children and I used to love to read. It's called Days Like These by Simon James. And this was always my favourite poem. And foolishly, the first time we read it, I went, oh, pick me. I know the answer to this. And of course, children being children, every time we read the poem after that, they waited for me to go, oh, pick me. But the answer to the question, how do you suppose it stores up all of the things it knows, is deoxyribonucleic acid, easily known as DNA. So what is DNA? DNA is found in all the cells of our body. Okay, Gareth, I know there's an exception, but, most of the cells in our body. It acts like an instruction manual that tells our cells what to do and how to behave. And your DNA is unique to you, and it contains all the information to make you, run you, and repair you. And the only exception to that is if you happen to be an identical twin. What is DNA made of? Well, it's basically two strings that are joined together with these molecules called nucleotides. And there are 4 different nucleotides in DNA, which I'm just going to call by their letters, A, G, T, and C. And the genius of DNA is that these nucleotides can join together, but they can only join together in specific pairs. So you can join a C to a G, and you can join an A to a T. And so it means that one strand of the DNA has got a code GAT, the one that is opposite to it or complementary to it will have the code, I've got to think, what did I say? GAT, so it would be C, my brain's gone, but it would be the other way round, the pair. So let me get this right. C goes with G, so GAT would come out as ATC. So DNA is confusing. But the genius, and that is the genius of DNA, this double strand with a complementary code running through it. Because every time you make a cell or you make a baby, you have to copy your DNA to put into that cell or to put into that egg or that sperm. And so that double helix, that double strand unzips, and then the very clever mechanism of DNA can then use that strand as a template to make the next strand that should be alongside it, as you can see in this picture, so that one strand of DNA, one double helix, becomes 2. It's copied itself. And that is the genius of DNA. 2 strands with a complementary code, and there is only one way that that code can be copied accurately, and so you can make these copies of your DNA. Now, the body reads DNA, or in this case, a molecular scientist reads DNA like a code, just like you and I might read information in a book. And the part of that code that we know most about is little packages of information called genes. What is a gene? A gene is a length of DNA that instructs a cell to make a specific protein. And proteins are molecules that are key components of living organisms. In other words, the DNA, the genes, are the recipe to make a human body. Now, I'd just like you for a moment to imagine that this chocolate crispy cake is a living organism. It's a very simple living organism made up of 3 proteins. It's made up of crispy, it's made up of chocolate, and it's made up of mini eggs, okay? So this living organism has 3 genes. It has the recipe for making crispy, it has the recipe for making chocolate, and it has the recipe for making mini eggs. And you put them together, and you've got our organism. My crispy cake had 3 genes. The human body has approximately 20,000 genes. And we have 2 copies of every gene, one from mum and one from dad. But those genes, those recipes for protein, actually only make up about 2 to 3% of human DNA. If you imagine, I'm switching metaphors slightly here, if you think of a gene as an instruction book, within each cell of our body, those books are contained on structures called chromosomes. A bit like books in a library are contained on bookcases. And I chose this picture of a library because there's a double set of bookcases. And we have a double set of bookcases, one from mum, one from dad. And all the DNA in our entire library is called the human genome. That's a word that's around a little bit in the press. I don't know if you've come across it. But the word genome means all the DNA in one cell. So what is a genome? A genome is all of an individual's DNA with all the genes and the bits in between. And we don't know an awful lot about the bits in between the genes, but we do know that it seems to have an impact on function on determining where a gene is used and when a gene is used. In a genome, there's something like 3 billion bits of information. And for those of you that are into IT, that's about 200 gigabytes of data. I don't know if that means anything to you. Our bodies contain a lot of DNA. So if I took all the DNA in one cell of your body, it would be 2 meters long, which is this length. Can you see that? That's just from one cell in your body. And you don't have millions of cells in your body. You don't even have billions of cells in your body. You have trillions of cells in your body. So in the whole of your body, if you've got trillions of cells, all with 2 meters worth of DNA in them, that's a lot of DNA. In fact, it's enough to reach from the Earth to the moon and back 6,000 times. It's a lot of DNA. And how does it all fit into a cell? That string, that DNA, it coils up really, really, really tightly to form the chromosome structures, and then it's only unwound when the body needs to use it. But it does mean that chromosomes themselves, who are this tightly wound, densely packed DNA, are relatively big structures. We cannot see a single human gene with the human eye. But with the use of a microscope, you can see chromosomes. So remember, chromosomes are the structures that hold the genes. And this is a picture of chromosomes. Chromosomes in the cell are not this neatly arranged. This is the cell after it's been organized. But you can see that there are 2 copies of most of those chromosomes. Chromosomes 1 is the biggest chromosome by international agreement, Chromosome 22 is the smallest, and we all have, or should have, 2 copies of chromosomes 1 to 22, and then the 23rd chromosomes, you might remember from your school days, are the sex chromosomes, and they're given letters to distinguish them from the others. And this is a set of male chromosomes, male with a little m, they're not mine. And there's an X and a Y chromosome. So when a couple have a baby, they each pass on 1 copy of the chromosome, and that's how those genes you get 1 gene from 1 parent. And you'll see that males have an X and a Y, but females have 2 copies of the X chromosome, 1 from mum and 1 from dad. Now, every time you make another cell or you make a baby, you have to copy your chromosomes, you have to copy your DNA. And as I said, you unzip your DNA, you use a complementary strand to make your copy, but we know that copying mistakes occur. We know that every single one of us has mistakes, has changes in our DNA that are not present in either parent. And for most of us, those changes fall in places where we don't notice them. They just account for the normal variability between people. So I'm going back to my crispy cake organism. He's good. He's good. You'll remember that this organism's got 3 genes: crispy, chocolate, and mini eggs. Well, this is also a very viable and fully functioning crispy cake. It's just got some variations in its genes. The crispy happens to be Rice Krispies, not cornflakes. The chocolate is white chocolate, not milk chocolate. And the mini eggs are pink and blue and not whatever color they were, green and blue, I think, in the other one. But, my children would eat these just as happily as they would eat the other ones without complaint. So the changes in the gene here are just normal variation, not a problem. Okay? And normal variation is glorious. But this crispy cake has a problem in its recipe for mini eggs. Now, that's not too big a problem. It's a perfectly viable crispy cake. My children would eat it completely happily. They might moan a bit, where's the egg? But it's a great crispy cake. But this one has a major problem. This crispy cake has a problem in the recipe for crispy, and it's just not a viable crispy cake. So you can have changes that just make normal variation. You can have changes that cause a problem, but you're still a fully viable human being. Or you can have changes in your genes that just make it not possible for you to exist as a human being. So my job as a clinical geneticist is making a diagnosis. I'm a doctor. I make diagnoses. That's what I've been trained to do. And then I work with others to make sure that patients and families get the best care possible given the diagnosis that we have made. And the other really major part of my job, and the part of my job that I really love, is walking with patients and families, giving them complex information in a way that they can understand so that they can make an informed choice in whatever situation they've come to me in. And that may be a different amount of information for different people. Now, the way I make a diagnosis is essentially through pattern recognition. This picture and the pictures that follow are not pictures of my patients. They are pictures that are freely available on the internet. I want to make that clear. But I should imagine that most of the people in this room, if not all of you, could probably make a genetic diagnosis in this young lady. And whether you realize it or not, you're making what we call a Gestalt diagnosis. Because what you are doing is recognizing a certain set of characteristic physical features in Down syndrome. And your brain is not saying, oh, that person's got a very round face and a certain shape to their eyes and a slightly large tongue and a particular body shape. You just look at them and think, oh, that person's got Down syndrome. Because it's a pattern that is familiar to you. Down syndrome is a very common genetic condition. It happens to be caused by a whole extra copy of chromosome 21. And I don't know if you can see here, but all of the chromosomes, there's 2 copies of them. But if you go down to chromosome 21, I don't have a pointer, you will see that there's a whole extra copy of chromosome 21. And what happened was that when the egg was made and the chromosome was copied, and it should have just split off 1 copy of chromosome 21 to put in the egg, that splitting didn't happen. And 2 copies of chromosome 21 went into the egg, and 1 copy of chromosome 21 came from the sperm, and so the baby ended up with 3 copies of 21. Now, there's nothing special about 21 in sense of getting 3 copies of it. You could get 3 copies of any of the chromosomes. But what's special about chromosome 21 is that a baby with 3 copies of chromosome 21 is viable. You can survive with 3 copies of 21. You cannot survive with 3 copies of chromosome 2. So there's something about the genes on chromosome 21. Maybe it's the size of the chromosome, maybe it's the actual genes involved. There's lots of research going into it. But you can actually survive with that extra information. Now, this young lady also has a chromosome problem. She doesn't have a whole copy of an extra chromosome. She has a tiny piece of a chromosome missing. And she has a genetic condition called Smith-Magenis syndrome, which Clive mentioned in my introduction. Through a happy coincidence, if you like, I got involved with the Smith-Magenis Family Support Group, and that's been a very richly rewarding partnership with me for many years. Now, I would recognize someone with Smith-Magenis syndrome because I have a particular interest in that syndrome. But actually, many of my fellow geneticists might not. They might look at this young lady and think, well, she's got something. I think we should do a chromosome test. But they might not know exactly what she's got because her gestalt is not as immediately recognizable as Down syndrome and some of the other syndromes that we see. But where a geneticist might make the diagnosis in this particular young lady is in the pattern of her behavior. Because in Smith-Magenis syndrome, you make the sleep hormone, melatonin, when it's light instead of when it's dark. So you and I go to sleep when it's dark because we make a sleep hormone called melatonin. But people with Smith-Magenis, their melatonin production is the wrong way round. So they don't make it at night, and they make it during the day. So essentially, if you have Smith-Magenis syndrome, you are permanently jet-lagged. They also have a particular issue with impulse control. And that means that for various reasons, they have a particular problem with self-harming behavior. And it can be quite extreme self-harming behavior. And that combination of a parent coming in and saying, my child, they want to sleep during the day. I cannot get them to sleep at night at all. and keep trying to pull their teeth out might make a geneticist think about Smith-Magenis syndrome. So we look at pattern recognition of physical features, we look at pattern recognition of behavioral features and learning problems, but we also look at patterns of investigations. So if you met this young lady, she doesn't have a particularly unusual physical appearance, she doesn't have any particularly striking behavioral traits, she does have a little bit of learning difficulty, she has epilepsy, she has problems at the back of her eyes, and she has problems with her kidneys. But the reason I can show you this picture is because this has been in the national press, and this young lady was the first diagnosis from a national project called the 100,000 Genomes Project that was run in the UK to try and make genetic diagnoses in people. Now, when I was a medical student in 1987, I did my first genetics degree, I wrote a finals paper on the pros and cons of sequencing the human genome. It was a theoretical suggestion at the time, and there was a big debate about whether it was going to be worth the resources, both in terms of money and in terms of time. You'll be interested to know that in my essay, I was for it. I thought we should do it. The project started in 1992, I think, certainly in the early 1990s. It took a 10-year international collaboration for us to sequence the whole 3 billion letters of the human genome. And the first draft of that was published in the early 2000s. Now, today, in 2023, I am using whole genome sequencing on a daily basis. I can order a whole genome sequence in a patient. The data from that will come back to my lab within 48 hours. The analysis, on the other hand, takes a year. The trick to the analysis of the human genome or any genetic test is to try and work out this change that we can see in the genes. Is it just one of those normal variations that accounts for the normal differences between people? Or is it actually causing a problem in this patient? Tale of two cities. It was the best of times, it was the worst of times. It was the age of wisdom, it was the age of foolishness, it was the epoch of belief, it was the epoch of incredulity. I don't know why I chose that sentence. This is a normal variation. The O has been replaced with a U. It really doesn't matter. You might smile if you were an English teacher marking an essay where a student had spelt worst with a U instead of an O, but really, does it make a lot of difference? No. I can read that sentence out and you know exactly what I'm saying. But if instead of that O being replaced by a U, the O was missing, but the spacing of the letters remained the same, you'd get this. Complete gobbledygook. And it's the same in genetics. You get changes that make no difference, and you can get what we call nonsense mutations that make it completely unreadable and mean that that recipe cannot be used to make whatever protein it should be being used to make. And both of those are quite clear examples. My lab would say, oh yeah, that's obviously a normal variant, and this is obviously a nonsense mutation. That's quite clear. But what if it was just the phrase, it was the worst of times, that was missing? The whole phrase. So you get, it was the best of times, it was the age of wisdom, it was the age of foolishness, it was the epoch of belief, it was the epoch of incredulity. Have we lost sense there? Is that an important change? Does it actually make a difference? And those are the kind of discussions that we have with our lab. So they will come to me and they'll say, we found a change in this gene, but we're not sure whether it's a normal variant or whether it's a problem variant. And we'll sit and we'll look at it and we'll try and work out what effect it has on the code, but we'll also look at the patient and say, well, does what the patient presents with fit with what we would expect if you couldn't make this protein? Has there been anybody else described in the medical literature with this change in this gene or something similar? And so we work forward trying to find the diagnosis in our patients. And so my job is right on the edge of research. I'm not a researcher, but it's right on the edge of it. And one of my professors used to say, a clinical geneticist stands in the gap between a researcher and a patient. We're in the middle, trying to move things forward. So we look for pattern recognitions that are physical. We look for pattern recognitions in behavior. We look for pattern recognitions in the genetic code. And the last place that we look for pattern recognition is in the family. Now, this family tree represents a family with a history of middle-age onset deafness. The squares represent men. The circles represent women or girls. The circles and squares that are filled in are the individuals who have this deafness, okay? The ones that are not filled in are either not affected or too young for us to know yet whether they're going to become deaf in middle age. This tells me two things. First of all, it tells me the way that this deafness is inherited in the family, because it's been passed from father to children. So you have 2 copies of every gene, one from mum, one from dad. This deafness only needs a mistake on 1 copy of that gene to happen. And that's called dominant inheritance. And it also is really important for me to know, for reasons I won't go into, that it can be passed from father to son to confirm that it's dominant deafness. The other thing that tells me is who in this family needs to be monitored for deafness. So the person with the arrow, you might think needs to be monitored for deafness because their father's deaf and both their siblings are deaf. If you look at the gentleman on the left-hand side of the screen as you look at it, he's deaf. His children will need to be monitored. We don't know if his granddaughter needs to be monitored yet because we don't know whether his son is going to develop deafness. But if you look at the lady on the right-hand side of the screen, she's actually married to a gentleman who had 4 children by his previous wife. She doesn't have any children of her own. So though she very much considers those children to be her own, they are not at risk of deafness because they're not genetically related to her. Now, I also happen to know that the lady in the middle, her 2 children, are not at risk of middle-age deafness. And the reason I know that is because this is my family tree. And that arrow points to me. And whether or not I want to accept it, I am well past middle age. Both my siblings have hearing aids, and I don't. I'm very blessed. I have not inherited the family deafness. And so I'm not worried about my children being deaf. But the reason that I wanted to end my DNA slides with my family tree is just to talk a little bit about me. And this is really difficult for me to do. and I'll tell you why. I became interested in genetics as a medical student. I had the privilege of sitting in with a clinical geneticist at St. George's, and I knew that that was what I wanted to do. But I faced opposition. When I told people in my church at that time, it was not St. Paul's, I got, "You don't want to do that. You don't want to do that. You might have to have conversations with people about abortion." There's no might about it. I would. You will be faced with some really, really difficult ethical situations. I don't think you should go there. And one gentleman, a fellow medic, told me straight out that I'd sold out and that as a Christian doctor, I should be on the plains of Africa as a surgeon. The fact that I faint when it's hot and faint when I'm standing up was not something that he bothered to find out. I would be a disastrous surgeon on the plains of Africa for many reasons, but that is one major one. But also, people were needing care in genetics. But I just stopped talking about what I did in church. And that's why everyone said to me, "Why are you nervous about giving this talk? You talk about DNA all the time." And I do. But I don't talk about DNA in the context of my faith, because for many years I just did not mention what I did at church. And when I came here, people knew I was a doctor. People might have thought I was a pediatrician if they knew I worked at Great Ormond Street. They might have thought I was a GP. But for many years, I didn't talk about what I did. My other problem was not talking about it at work, not talking about my faith at work. All the controversy with the church and the debate about evolution had given people the impression at work that Christians were just idiots who weren't interested in looking at scientific information. But also, there was an experience with evangelical Christians. I'm going to be honest with you here. I went to a conference quite early on in my career where a genetic counselor, that's one of the associates that I work with who doesn't do the diagnostic part of my job but does do the information sharing part of my job, stood up and talked about it. There was a thing about genetics and faith groups, and she stood up and gave a talk and said the faith group that she found it hardest to work with was evangelical Christians. And the reason being was that when she came across evangelical Christians who had genetic disorders in their family, they would pray that God would rescue them from that genetic condition, and that then didn't happen. And then their faith fell apart, and she did not know how to handle that. And I sat there in the audience, sort of creeping down in my chair, thinking, how have we got here as a church? If your mother died of an inherited form of dementia and you are not a Christian, you have a 1 in 2 chance of inheriting that dementia. If your mother died of an inherited dementia and you are a Christian, you have a 1 in 2 chance of inheriting that dementia. We are subject to the random chances of genetic inheritance the same way as everybody else. I don't believe that our God steps in and rescues us from that. I do believe he walks with us through it. But I stopped talking about what I did at church, and I stopped talking about my faith at work. Next slide, please, Chris. Not surprisingly, that wasn't a great strategy. And after several years, I ended up in a really angry and sad place. I was angry about what I was seeing at work, We talk about people being fearfully and wonderfully made, and I was seeing babies who definitely weren't fearfully and wonderfully made. But I couldn't talk to anyone at church about it. I didn't even talk to God about it because actually I was really scared that he didn't approve of me doing clinical genetics either. And I did not know where to go with this grief. By that time, I was here, and there were a couple of people in this congregation. One was a very, very good friend who held me in my anger, didn't try to fix me, didn't try to tell me what to do. She just held me in my anger and prayed for me and let me weep. And then another was a very wise Christian counselor who actually, sort of said, well, who told you God didn't want you there? But also just gave me some techniques for dealing with grief. So she actually took me on a grief walk through my medical career so that I could just speak out the pain, and then gave me a practice of when I'm on that train coming home from work and I'm really, sad about a situation, to actually look at that situation and think, well, what is mine to do? What is my job to do in this situation? And then everything else on that train journey, to just let that go, leave it by the tracks, if you like, leave it with God. I also read a book, and I tried to find the book before we came out, and I couldn't find it. I must have lent it to someone. It was a book called The Good and Beautiful God, written by someone called James Bryan Smith. And in a chapter called God is Good, Bryan talks about the loss of his child with a chromosome disorder. I was like, okay, God, okay, this chap has been through what I've only watched. He's been through it, and he can say that you are good. Right now, I can't say that. Right now, I don't really want anything to do with you, but on the basis of Bryan's testimony, I'm prepared to pray, give me the desire to desire you again and help me to see that you are good. And that good friend and my husband and her husband worked through that book, The Good and Beautiful God, and it was life-changing for me. And I then went on to study with an organization in the States called Renovare, which is an organization that is about knowing God better, is about us living a with God life. It's about us not just talking about our faith, but making our faith active in our lives. And through that course, I was really challenged to try and bring the two halves of my life together. I was really challenged to be brave and tell people at work that I was a Christian. I was also really challenged to be brave and tell you lot that I'm a clinical geneticist. So be gentle with me, please. And the last slide, please, Chris. I haven't answered the questions. I still look at God and say, I truly believe that you created via evolution. I think DNA is genius. I think there's something about randomness in there that is connected to free will. God's hands off, giving a space where free will can happen. But I don't understand all the answers. I'm not a theologian. I cannot explain to you why a child is born with a neurodegenerative condition. I can't do that. But what I have learned in the last few years is that God weeps when I weep, and he weeps with the families that are suffering. What I've learned is that Christians, we need to be in there. We need to be in the conversation. you know, yes, I talk to people who are making difficult decisions, but with every ounce of integrity that I have, I give them as accurate information as I can so that they can make an informed decision. It's their decision, but I want it to be an informed decision. And I really do believe that God is in genetics well before I was. But I want to walk with him in my life there. And to do that, as I said in the article in News, I need wisdom. I need prayer. I need the Holy Spirit. I do believe that God is light, and in him there is no darkness at all. But there are dark places in this world, and I want to I want to be his light there. I want to be a conduit for his light there. I think I'll leave it there. Thank you very much. I'm happy to take questions.
Any questions? There we go. Let me come over so that we can use the mic, please.
Annalisa was first, I think. Annalisa was first up.
Okay. Where are we? That's all right.
Hi, Annie. Just wondered if ADHD and autism comes under the chromosome being wrongly copied.
Okay, so both ADHD and autism have a genetic component, but neither of them are usually caused by a problem with a single gene or a single chromosome. They are both what we call multifactorial disorders. So there may be several genetic factors coming together, and there may be environmental factors and other factors as well. There certainly isn't a genetic test for ADHD or for autism. But we do know that certain genetic conditions are more likely to be associated with autism and ADHD. So they're both very complicated conditions, not very straightforward, I'm afraid.
Who's next? Yeah.
Hello. We know about Down syndrome, and you mentioned Smith's syndrome.
Smith-Magenis syndrome, yes.
Are there many just conditions that are just one, not like autism, but just one, caused by just chromosome being wrong?
Just one thing.
Yeah.
Yes, there are thousands.
Thank you.
There are thousands. And probably, if we knew about it, there's probably at least 20,000. It's just that we don't yet know what all the genes do. So there's still a lot of people out there that we suspect have a genetic syndrome because there's something about their physical features or the pattern of inheritance in the family. There's something that, or the picture of their MRI scan that points to a problem with a gene, but we don't know what that is. And the 100,000 Genomes Project was set up in this country to try and take those diagnoses forward, and now that's gone into clinical practice, where we are trying to find the answers. But even then, our knowledge is such that, I have patients that I have had a whole genome done, and I'm sure they've got a single gene disorder, but that we've not found it yet. And that will be a limitation on our knowledge and still our limitation on our analytic techniques. So we'll get there one day, but long after I've gone. Yeah.
Hello, thank you for the speech. Regarding epigenetics, how does trauma fit into this? Is there data to suggest that trauma might be inherited over generations? And if so, how does that work as a mechanism?
So I think you're asking me about epigenetics, is that right? So what I've been talking about is structural problems with the DNA itself. But DNA is obviously more complicated than I have told you about. And there are mechanisms for switching on or off of genes. So one particular example is growth genes. There are certain growth genes that if you inherit them from your father, they're switched on. And if you inherit them from your mother, they're switched off. And the sort of, the argument for that is, if you like, is that if you're a man, you want your baby to be strong and survive. But if you're a woman, you want to be able to give birth to that baby without dying. So we're all a balance of the genes that are switched on and off depending on where they come from. But I will have had genes that were switched on when I inherited them from my father that were then switched off when I passed them on to my children because I'm a woman. So the changes in those genes are not permanent. They are changes due to other molecules other than DNA that are stuck on the DNA that prevents or allows that gene to be read. It's very complicated. And there are various, it's very much a subject that's still being investigated, but there are various studies that can show that periods of famine or periods of immense stress in a population can then affect the genes that are switched on or switched off to be passed on to the next generation. And some of it sort of makes sense, so that if you've gone through a period of famine, the next generation might have more tendency to be obese because the genes are switched on that allow you to kind of get every bit of nutrition out of the foods that you eat. But then if there isn't famine in the next generation, that's not necessarily needed, and therefore you might become overweight. But it's not my field, I think is the first thing to say. And the second thing to say is I think it's still a very young field that we're still learning about. But some of the single gene disorders that I deal with are genes that control that epigenetics, which then is a permanent gene change that affects the temporary gene changes, and it's all very complicated.
Hello, I'm Marcella. Thank you for your wonderful talk. I would like you to suggest a book, just a basic, simple book, to learn about what the topic you've been talking about, because, it would be nice to have a read as well as,
Yes.
Thank you.
Certainly. Well, I do actually think this is a good book. It does go into very basic DNA biology. But otherwise, any good GCSE biology textbook is a really good place to start. I don't know if you're familiar with the internet, but Chris, I don't know if you've still got my slides up, but there was a slide of the reproduction of DNA that, I can't remember the exact website, biologydictionary.net, which is an open access educational website about all sorts of biology, but it has information there about DNA and how it replicates. So I think you could start in any of those places.
Anybody else? Yes, Sarah.
Thank you. I was just wondering, in all the work that you do, how much through what you learn is you're able to help couples avoid getting some of the DNA problems in the future. I remember back when we were starting a family, I was told to have folic acid to try and prevent Down. So I just wondered if there were other things that you can find within what you do that help people, at least going forward.
I think that's a really, really good question, and I think that the folic acid thing is a really important thing because it just illustrates that not every problem that a baby has is due to a genetic problem. So lack of folic acid actually predisposes to a condition called spina bifida. And in this country, we recommend that any woman who is wanting to get pregnant takes folic acid for at least 3 months before pregnancy and 3 months into pregnancy, because it's lack of folic acid that makes spina bifida more likely. But also we know that smoking makes babies very small. Alcohol increases the risk of ADHD and learning problems. So there are many viruses can cause problems. There are women who were pregnant early pregnancy when they got COVID, and that's affected the growth of their baby's brains. So, there are very many different things that can affect the health of a baby. And one of the things I often deal with is when a couple have had a baby with a genetic problem, they are very understandably terrified about having another baby, about what's going to happen next. And they want me to give them the test that will prove that they've got a perfect baby. That test doesn't exist, but also even if it did and I could prove that a baby was perfect, that baby, you're going to think I'm really negative here, but I'm just trying to make a point. That baby could get meningitis at day 2 of age. Life is not purely about genes, and you cannot be a parent without risk. It just is not possible. But obviously, if there is a genetic condition in the family, then there are things that we talk through with a couple about the next pregnancy. So one option is not to extend their family. One option is to consider adoption. One option is to consider sperm or egg donation. With some conditions, but not all, it's possible to consider a technique whereby you can test an embryo for a genetic condition before it gets implanted into the mother's womb. And then for some couples, the option is whether or not they will, to test a pregnancy and to decide whether or not to continue with an affected pregnancy. And that's a conversation that I have with people very, very regularly. And I think if there's one message I would like to get across to the church is, please don't judge. Please do not judge. The pregnancies that I'm talking to people about are usually very much wanted pregnancies. Nobody takes these decisions lightly. The families usually know what this condition is like because often they already have a child with that condition. And they know whether they can cope with another child with that condition or not. They're very, very difficult decisions. One thing I would say to you is pray. Pray for the families in that situation. Pray for those of us that are advising them. But also give thanks for the fact that in this country we have a very, very ethically minded genetics and fetal medicine service. The ethics that they have might not exactly be the same as yours, but I can assure you that human life is very much valued. And people take conversations about whether or not it is appropriate to offer a genetic test in pregnancy or whether it is appropriate to offer a termination of pregnancy very, very seriously. I have never ever been in a situation where someone's just said, oh, just get rid of it. I've never been in that situation. The people that I work with really want to get things right. And you may be someone who thinks that abortion is absolutely wrong in every circumstance, and absolutely you are, that is a valid opinion. But please don't think that because someone thinks slightly differently from you and thinks that abortion is acceptable in certain circumstances, that they don't care. Please don't think that they're not ethical and that they don't value human life, because my experience is that people do. And one of the things that I mentioned was that I'm specialist advisor to the HFEA, and that's the Human Fertilization and Embryology Authority. And we should give thanks that that authority exists. It is an organization that is set up to monitor fertility services in this country and to monitor what is done to human embryos. And honestly, any handling of human embryos in any lab in this country, whether it's a research lab or whether it is a fertility lab, is monitored and regulated by the Human Fertilisation and Embryology Authority, and they take it very seriously. So pray for them. Pray for them that they make right decisions. Pray for them that they will appoint wise people to their board. And my job, I act as specialist advisor to their licensing committee. So if you wanted a genetic test on an embryo in this country, that genetic test would have to be licensed by the Human Fertilisation and Embryology Authority. And they have criteria. They will not allow you to have a genetic test just to tell you whether your embryo is male or female. That is absolutely out. They will not let you have a genetic test for any minor condition. It has to be, in order to muck about with human embryos, it has to be a significant genetic condition and a significant risk of that condition occurring in that embryo. And my job is to give them the facts when they make those decisions. So somebody puts in a license application for a particular genetic disorder. My job is to review that license application, to tell them whether the facts in that application are true, to tell them the inheritance of that condition, to tell them the severity of that condition, and to answer any questions that the non-geneticists on that panel might have about that condition, in order that they can make a decision that is in line with the very strict guidance that there is in this country. That is not true across the world. There are some countries where you can have testing for anything. So give thanks for the fact that we have an ethically minded country that cares, really cares about human life and treats it with respect. And pray for the people that are involved in those decisions, please. And pray for the fetal medicine units. You know, so the fetal medicine doctors are obstetricians who look at babies under ultrasound and look for scan abnormalities. I have never, ever met a fetal medicine consultant who wants to do an abortion, ever. It's a horrible thing for them to have to do. But they do it because they think that it is the lesser of two evils in that situation. You might not agree with their decision in every case, but please pray for them because they're trying to make the right decision.
Okay.
If you didn't care, you wouldn't go into it. Sorry.
That's all right. No, don't
get off my hobby horse
don't
now.
you dare apologise, because I think a lot of what you've just said has been very reassuring and opened our eyes a bit to more of a global view rather than just what we feel inside. And thank you so much for coming today. Let's
Let's see.